on to one's surroundings, a behavior that implies the existence of a brain and a nervous system. I'm your host, Bruce Lipton, for this series on inner evolution. Convention holds that the genome-containing nucleus represents the cell's brain, for it was perceived that genes turning on and off would control the cell behavior. As described previously, genes are not self-actualizing. They are simply blueprints that are read to reproduce the proteins and the cell itself. Consequently, the nucleus is actually the cell's gonad. The brain of the cell is the cell membrane, for it is this structure that reads environmental signals and elicits the biological responses that control behavior and genetic activity. The structure and function of the cell's membrane reveals that it is an information processor, a carbon-based chip that provides the interface between the mind and the body. The separation of the mind-body duality is actually first attributed to René Descartes. Now, Descartes was a philosopher in the early ages of the era of modern science. And back in 1637, in his book on the methods of science, he wrote this following phrase, and it is, cogito ergo sum. I think, therefore I am. And what Descartes was really describing in this was the fact that knowledge up until this time was just based on hearsay, that there was no science to understand what was actually going on. And that René Descartes suggested that we get rid of all previous knowledge and start to build a new knowledge base based on the method of science. His only truth that he could start with is that he existed, his mind, I think, existed, and the body existed. And in creating cogito ergo sum, what René Descartes was actually saying is that mind and body represent separate and distinct substances. The mind was a thinking substance with no physicality to it. In contrast, the body was a physical substance with no thinking attributed to it. So we separated mind and body very early in the era of modern science. And yet, this separation continued 50 years later when Isaac Newton wrote the Principia, the founding books of what is today called classical physics. And in that physics, what Newton did was separate mind and body just like René Descartes did. And he separated the mind and the body based on a simple reality. According to physics, the universe is comprised of two elements, a physical material element and an invisible energy or spiritual element. Well, the reason for separating mind and body in Newtonian physics is simple. The body is physical, the mind apparently is a non-physical energy element. But there was a little bit more to that separation as well. At the time Newton was creating classical physics, civilization was actually being controlled by the church. And this separation allowed science to exist outside of the realm of the church. Well, the whole idea of mind comes back into our reality in the era of quantum physics. Max Planck in 1944 wrote, "All matter originates and exists only by virtue of a force. We must assume behind this force the existence of a conscious and intelligent mind. This mind is the matrix of all matter." Well, this becomes really important in understanding the reality of our world, because the mind now is an energy that is recognized by quantum physics. And this returns mind back into the story of science, so we can engage mind with the physical realm. And this is very interesting because Albert Einstein followed up with a very important quote that we had mentioned earlier. That quote is, "The field is the sole governing agency of the particle." Why is this relevant right now? Well, the answer is this. The mind is an energy field. The particle is the body. So if I repeat Einstein's quote, putting mind and body in, it will read as follows. The mind is the sole governing agency of the body. And this statement brings the mind back into our understanding of science and its role in influencing the character of our lives. We describe the nature of biology using the model of a simple machine. The machine consists of a mechanism. There's a force that drives these protein gears, and that was the signals from the environment. In our model, we also have a switch that turns the motor on and off that actually would control behavior. So what I'd like to describe right now is the nature of that switch. The switch that invites a signal from the environment to control the behavior and genetics of the cell. So we go back to a very simple equation about life, and that equation is this: protein plus signal equals behavior. Well, the proteins are in the cell, but the signal comes from the environment. For the signal to cross into the cell, it has to pass through the cell membrane, and that's the skin of the cell. So to understand how signals engage biology, it is important for us to understand the nature of the structure of the cell membrane and how that structure will facilitate an environmental signal crossing into the cell and controlling the behavior of that cell. Well, the first thing about a membrane is this. It was only in the late 1940s that biologists even recognized that cells had membranes. And the reason is this: the membrane is so thin, it cannot be seen with a regular microscope. The membrane, to be seen, we had to use an electron microscope, and that was developed in the 40s. At the first time cells were observed, along the outer surface of the cell was a layer, the membrane. There were 3 layers to the membrane as seen in the electron microscope: dark, light, dark. Now, what's really significant about this identity of the membrane is this. In biology, structure relates to function. The more complex the structure, the more complex the function. Well, when they looked at the cell membrane with a simple trilaminar layer, they said, well, there's not really much structure to that, and that all of a sudden implied that perhaps there wasn't very much function to the membrane. In fact, scientists perceived of the membrane as nothing more than plastic wrap containing the cytoplasm of the cell. It's just basically a structure that holds the cytoplasm together. Well, that was based on the electron microscope. The simple trilaminar structure of the cell membrane actually reflects the molecular organization that is the foundation of this membrane. The membrane is composed of a bilayer of what are called phospholipids. Phospholipids are very interesting molecules because half the molecule is hydrophilic, meaning it would love to be dissolved in water, but the other half of the molecule is hydrophobic and avoids water. The significance of this is that the molecules forming the cell membrane create a layered structure with lipid in the center. The membrane is a barrier, but the lipid in the center resists water. It's hydrophobic. So what that means is anything from the outer environment that is soluble in water cannot pass through the membrane because of the lipid center, which will resist the flow of water molecules going across the cell. So the significance is this: the membrane structure, by definition, is a crystalline structure with a lipid core that actually makes it a non-conductor. Trilaminar structure I can illustrate by using an analogy of a bread and butter sandwich. Now, the bread on each side would represent the phosphate groups because bread, like phosphate groups, can absorb water. But in the center of the bread and butter sandwich, the thick layer of butter is the equivalent of the lipids in the center of that membrane. Now, the issue about this is if we tried to feed the cell some food in our model, so let's take a spoonful of grape juice and drop it on the bread and butter sandwich, the question is this: will that food enter into the cell? Well, if you section this bread and butter sandwich, guess what you find? Yes, the phosphate groups would absorb the food, but the food would not be able to penetrate through the lipid layer in the middle, and therefore nothing can get into the cell, and in fact nothing can get out of the cell because the membrane at this point is simply a barrier with a hydrophobic core in the center of it. Yet, the membrane is responsible for allowing food to come in and waste products to come out, as well as signals to come in. I said, well, how can this occur in a bread and butter sandwich? And the answer is this: that what was not seen in the electron microscope in our picture of the crystalline structure of this membrane is the fact that there are proteins built into the membrane. And they're built into the membrane, hence the name of these proteins are called integral membrane proteins, abbreviated as IMPs. Well, there are two major classes of IMPs built into the membrane. One of these protein classes is called a receptor. I say, what are receptors? Well, these are proteins that respond to environmental signals. And it's very interesting because our skin is analogous to the membrane of the cell. And where do you think our receptors are? Eyes, ears, nose, taste, touch, pain, temperature, pressure. Like the cell, our receptors are built into the skin, the cell membrane. But when we go back to the cell membrane, the receptor is simply a protein with an antenna, and the antenna vibrates and resonates with signals in the environment. So a receptor picks up a signal. What happens to a protein when it complements with a signal? When a signal shows up that the receptor can read, guess what? The receptor changes shape. And now the receptor can connect to the second class of proteins. And this class of proteins includes channels. Now channels, as the name said, are proteins that have a conduit through it. In a resting state, a channel is closed, so nothing can get through that channel. But when the channel is activated by a signal, the channel opens up and allows the conduction from one side of the channel to the other side of the channel. To visualize what this actually represents, I have an animation. It's a piece of membrane cut out of a cell. In it, you'll see a receptor with an antenna and a channel. And at this point, the channel is closed. Now, what will happen is this. The receptor is scanning the environment for a signal. And when the signal shows up, it's going to cause the receptor to change shape. And when the receptor changes shape, it allows a protein called a G protein to connect between the receptor and the channel. When the G protein connects to the channel, that's the signal for the channel to open up. And when the channel opens up, it forwards a signal from the environment into the cell, a physical signal that will now control the behavior of the cell inside the cytoplasm. So this is actually a switch. It's a membrane switch. It responds to a primary environmental signal through the receptor. The channel creates a secondary signal, which goes into the cell to control its function. Thank you for joining me. I'm your host, Bruce Lipton, for this series on inner evolution through biological change. Join me for the next episode where we'll talk about the seat of consciousness.
8, 7, 6, 5, 4, 3, 2, 1, 0.
2020, the year of karma. 2020, the ultimate Red Pill.